describe here the Morphology Synthesis Colab, which allows you to create and analyze neurons. You arrive on the overview page. On the left of this page is the navigation pane. On the right, the collaboration pane, including a chat window where you can talk with other members of the Colab, ask questions and share ideas. In the central overview pane, you find a description of the synthesis process and some images of neurons that have been created. This figure shows 300 synthesized cells of mixed M types in a hexagonal column. And here are several cells that have been synthesized in the volume of a rodent hippocampus. After this description is a useful links section, including a link to information on how you can get access to the Blue Brain Project HPC resources that are needed to synthesize neurons in the Colab. And at the bottom, we will be posting publications related to synthesis. You can use the navigation pane on the left to move around in the Colab. For now, let's go to the Getting Started page that describes how to synthesize neurons. Here is the workflow for synthesis and validation of cells. There is one notebook for synthesizing the neurons and a second notebook for validating them. After this description, there is a navigation section which explains all the pages in the Colab. As well as the overview and getting started pages, there is a tasks page showing the synthesis and validation task windows, but this is not used in this demonstration. The storage page allows you to navigate to the files and explore the data you have produced. The job manager shows the status of the jobs you have submitted, both queued jobs, running and completed jobs. The members of the Colab are in the team page, and the settings page is used by Colab members to set preferences. At the bottom are the Jupyter notebooks where we will carry out this demonstration. Under the navigation, in the central pane, the synthesis and validation tasks are described in detail, including the input and output file formats, the kind of data needed to synthesize cells, and the type of cells that can be synthesized. Currently, there are 3 distinct initial configurations that specify different volumes of space and numbers of cells. New configurations will be added shortly. After the synthesis description is a description of how morphologies can be validated. At the bottom of the navigation pane are the Jupyter notebooks. Open the Morphology Synthesis notebook by clicking on its menu item. To execute this notebook, you must first have created a new empty directory on the Blue Brain Project GPFS file system to hold the output files. This can be any directory path to which you have write access. We would use one similar to the one presented here. In order to synthesize cells, you have to specify the initial configuration that is to be used. This is identified by a UUID here. If you return to the Getting Started page and go down to the table, you can then cut and paste the UUID for the configuration you want into the notebook. You should do this before executing the notebook. We can paste this in here. Note that a default configuration will be 20 cells produced in a hexagonal column, and you can use this if you desire. Now, replace new_dir by the directory that you want to use for this job, and specify a random number seed, which must be a unique positive integer, in this field here. Now you can proceed to execute the notebook by clicking Shift-Enter in each of the active cells. Once you get to the task wait cell, this is the synthesis execution, and it may take some time depending on the load on the server and the number of cells you have specified. In order to see whether your job is executed, open a different pane in the browser and navigate to the job manager page, and then you will see that your job is sitting in the queue and has not yet executed. Return to the original notebook and wait until the job executes in order to proceed. On completion, the synthesized cells will be available in the directory you specified on the GPFS server, and you can proceed to apply a set of validations to them by following instructions in the validation task notebook in the Colab. Or you can get the cells directly from the GPFS directory you specified earlier. You have just completed the morphology synthesis task.